So on September 27th, just this past week, we had a big meeting in Boston called Defining a Roadmap for SHANK3-related Epilepsy Research. And we know that this is so important to so many people in the community because so many people have epilepsy, and also we don't know, like none of us knows if our kid currently is fine, if they're going to be the next one to start having really big problems with seizures. So I am going to, for a moment, turn it over to Paulina Rychenkova. She is CureSHANK secretary, and she and Geraldine Bliss, the president, were there, and she's going to tell you all about it.
Thank you so much, Talia. I want to say that that Geraldine Bliss really drove the organization of this meeting. It was her brainchild, and she worked very closely with the developmental the SHANK consortium led by Mustafa Sahin. And so what I'm going to tell you a little bit about the goals of the meeting, what we did during the time that we all got together, and a few highlights and key takeaways, and I'll ask for Geraldine to weigh in with her comments afterwards. As Talia mentioned, this is CureSHANK's effort to identify opportunities for us to fund research, whether it's some basic research or some clinical and preclinical research that would allow us to understand better how epilepsy plays out in Phelan-McDermid syndrome. So CureSHANK funded this meeting. The meeting goals are outlined here. They were twofold. The first goal was to identify the key gaps and needs and challenges that hinder progress in understanding epilepsy and Phelan-McDermid syndrome. And the second goal was to develop a set of research priorities and recommendations that could create a roadmap for CureSHANK and other organizations to potentially fund. The meeting co-chairs were Mustafa Sahin from Boston Children's Hospital, Jimmy Halder from Baylor College of Medicine, and Liz Berry-Kravis from Rush. The agenda briefly summarized on the right. It was a very productive, almost a full day meeting that was attended by over 24 researchers. The researchers were both basic researchers from academic research centers, as well as clinicians, practicing clinicians who work with patients who have epilepsy, Phelan-McDermid syndrome patients who have epilepsy. So we kicked off the meeting by sharing family experiences, and we are very thankful to these individuals who either participated in person to share their family experiences with epilepsy and Phelan-McDermid syndrome, or actually recorded a video testimony that was shared with all the participants in the meeting. It was really important to hear directly from the families who are living through these difficult challenges. We then heard from Jimmy Halder, who provided a very comprehensive clinical overview of what is known about cases and instances of epilepsy, prevalence of epilepsy, the types of epilepsy that our children experience. The basic science overview was provided by Dr. Buxbaum from Mount Sinai, Seaforth Center at Mount Sinai. And then we split up, all the participants in the meeting split up for about 2 hours each into two groups. One group tackled clinical gaps and needs and challenges, and the other group tackled the basic science gaps, needs, and challenges. We then reconvened and shared what the discussions concluded with each other, or one another. The basic scientists and the clinical scientists could then ask each other questions that may have come up during those brainstorming discussions. And then ultimately we coalesced around kind of what the next steps are and started working towards setting those research priorities. Next slide, please. I wanted to share with you some of the key takeaways from the meeting. By no means this is an exhaustive list. There's a lot more to work through and digest from those discussions, which we are actively doing together with the co-chairs and some of the other participants of the meeting, because ultimately what we would like to do as the next step is for CureSHANK to develop a specific request for proposal that would go out to potential applicants, and we would fund that project with the $125,000 grant. So some of the gaps that were identified, and these are combined basic science and clinical gaps, include gaps in consistent and repeatable and reliable 2D cellular or organoid type models and animal models of epilepsy and Phelan-McDermid syndrome. We currently do not have reliable models at any level that spontaneously display seizures or epilepsy activity in Phelan-McDermid syndrome. It's important to have model systems to study any neurobiological disorder, and therefore, for us to understand epilepsy better and the mechanisms underlying epilepsy and Phelan-McDermid syndrome, we really need to identify models that are effective for this. Another gap is a detailed and in-depth knowledge of the SHANK3 gene. The scientists have a fairly decent understanding of the gene, but still there are significant gaps in that. And the reason it's important is because when companies start thinking about different approaches to treatments that are maybe more in the realm of gene therapy or mRNA type therapy or RNA type therapy, understanding every part of the gene is really, really significant. And there's some gaps in that. Another gap on the basic research side was really understanding the circuit phenotype, and what that means is it's epilepsy and seizure disorder does not really happen at an individual neuron level. It happens at the level of neuronal circuitry. So understanding the interplay between excitatory synapses, inhibitory synapses, and various other cells is really key for understanding epilepsy. And so we're still seeing there's a gap in that understanding that still exists. On the clinical side of epilepsy, some of the gaps that were highlighted were the need for a better natural history of epilepsy and Phelan-McDermid syndrome that includes onset, age of onset, types of seizures, the types of treatments that have been employed and have been effective or ineffective, maybe trying to capture some behavioral or other events that may have led to seizure development, or something that maybe at the time didn't seem significant, but in retrospect was probably indicative of seizures that would develop in the near future. Another gap is trying to understand why do some individuals with Phelan-McDermid syndrome ultimately develop seizures and epilepsy, and some do not. And another gap that we'll conclude here, and again, I want to highlight that this is not an exhaustive list, but another important gap on the clinical side is trying to ascertain whether there are any differences in brain structures between individuals with Phelan-McDermid syndrome who have epilepsy from those who do not. And as I mentioned, the next step for us is to really digest all the discussions and kind of review the gaps that were identified and come up with a well-formulated request for proposal. And at this point, I'd like to see if Geraldine would like to make any comments.
Oh, put me on the spot here. I think, there's been an incredible wealth of research on SHANK3 and on Phelan-McDermid syndrome over nearly the last 20 years. And most of it has really been focused on things like behavior and learning in Phelan-McDermid syndrome, not on epilepsy. And indeed, it likely stems from the fact that much of the research on Phelan-McDermid syndrome has been funded through pots of money that are set aside for autism-associated research. And we've had the good fortune of our field benefiting from that. And now we kind of need to work intentionally to attract funding from the National Institute of Neurological Disorders, which typically funds epilepsy research. We only have one NINDS funded research study that's ever been conducted. And so we need to see many more of those kinds of projects funded by NINDS. So our hope is that our initial funding, our $125,000 grant, will sort of serve as a seed grant so that our research community can go to the NINDS with good data and be more successful in applying for grants from the NINDS related to epilepsy.
Thank you. And with that, we will hand it back over to Talia. Thanks.